Welcome to the future of medicine. So we're going to particularly talk about a future of medicine with a vision that DNA becomes a really big part of that. And it's a credit to, I think, March Capital that they've been recognizing that the themes of generative AI are going to tackle much more than our tech realm, our industrial realm, but they're going to revolutionize biology. And in fact, where you might want to most revolutionize biology is at the level of DNA. DNA is the code of life. It instructs the natural world around us. It is the original information technology. And while we've made amazing progress in sequencing that code over the past 20 years, from the first human genome being sequenced to millions headed towards billions, we have made a lot less progress in reading that code, meaning actually understanding what those letters mean, and even less understanding in variations of writing that code. So Mike and Nicole are going to bring us into the reasons to believe that DNA is going to be a big deal. And if you were to add up the market caps of all companies in variations of DNA medicine today, it would be maybe 0.1 trillion with generosity. If you add up all the market caps of protein therapeutics, that's above $1 trillion. If you add up all the market caps of companies doing variations of small molecules, that's a few trillion. Zoom out a little bit into the future, and those are likely to be flipped. In that the best case scenario is that a medicine cures a disease or prevents disease from ever happening. And DNA is going to be the rightful home for many of those medicines. So we're going to go on a little voyage today and learn a little bit about that. I'd love you both to introduce yourselves and what led you to come to the conclusion recently or long ago that the future of genetic medicine was going to be a big deal. Nicole, do you want to kick us off?
I can start off. So my name is Dr. Nicole Polk. Up until a hot minute ago, I was actually a professor at UCSF in San Francisco. Wasn't out amongst all of you in the industry space. And decided to make the pivot here about 2 years ago to switch out into industry because I saw some data in my lab that was quite literally some of the most exciting data I had seen in about 17 years, and the thought of handing my baby off to somebody else to work on was unacceptable to me, and so decided to make the switch in 2021. And so we use viruses as medicines, right, which I think is a new concept. I think a lot of people only know about, if I polled most of you and said name a virus, there's probably only about 20 you can name, and those are the ones that make you sick. The vast majority of viruses that we've ever discovered on the planet actually don't make you sick. They don't make you sick at all, but they're still very good at getting inside of you, and so we can actually engineer these like little FedEx trucks to deliver any type of medicine, for the most part, anywhere in your body that we'd like. And so using viruses as tools to deliver genetic medicines, things like DNA, are where I've kind of scratched my teeth, and we've been working for about the last 17 years, and so that's, we'll start there.
Beautiful.
All right. So it's a pleasure to be here today. My name is Mike Severino. I came into this through a similar route. I'm a physician scientist by training. I've spent essentially my entire career trying to figure out how to understand and then ultimately treat human disease across a wide range of areas. I've been working in the industry for quite a long time, for the majority of my career. You know, the dirty little secret that we don't talk about a lot in medicine is that most of the interventions that we use have very, very modest effects. So we can use statins to reduce the risk of a myocardial infarction and heart attack. The absolute risk reduction from the best statin therapy is less than 1% today, okay? So that's good if that's all you can do, but it always struck me that we had to do better. And DNA, as Jeff said, is the code of life. It encodes the genes that are responsible for our health and disease. And by getting at the core of the problem for the first time ever in the history of medicine, I think we can do much, much better going forward.
Yeah.
Beautiful. So let me draw us into what you're specifically working on. So Nicole, you described that the trigger to jump out of academia wasn't bureaucracy or inefficiency, although you may be seeing
those contributed,
some of those
don't get me
benefits. But with some of the most exciting data you'd ever seen. Walk us into what is that data and how has it informed what you're trying to do in Siren?
Yeah, so when we first started my lab, we wanted to kind of whiteboard out and do kind of a red room exercise and think through what are all the biggest, gnarliest challenges that are going to have to be solved from within academia because they're going to take too long and be too risky. And in my field, which is gene therapy, where for the most part we use viruses to deliver genetic medicines to patients, Every single one of them that has ever existed on planet Earth, whether it's from academia, small startup, big company, big pharma, government, doesn't matter. Every single one of them is bespoke and personalized for a single disease, right? So you can't use a virus to treat a little boy who has hemophilia B with a virus you made for a little girl who has spinal muscular atrophy. Those diseases are different, and the payloads, the medicine that you're delivering inside of that virus is different. And so we're like, this isn't gonna scale, right? There's 8,000 single gene genetic disorders, and there's a whole lot more common acquired disorders, the types of things that many of us in the room either already have or will develop as we age. And we're like, how are we gonna scale this into a disease that has maybe 2 million patients a year, not 10 patients a year that get afflicted, right? Because making viruses is quite expensive. And so we were like, is there a way that you could make a virus that is not unique to just a single disease, but one that's universal, one that could be used to treat dozens, hundreds, or thousands of diseases with the exact same virus, the exact same drug? And so we kind of took a bioinformatic approach to that and thought through, okay, let's step back from these single gene disorders that historically my field has worked on and think more broadly, like, what are types of payloads that we could deliver inside these viruses that could do something of interest therapeutically for a prevention or a cure? And so we're like, got excited about things like transcription factors and cytokines and things that modulate protein degradation and cell fusion and all of these types of things. And got excited about, made this huge master list and decided rather than testing 1 or 2 or 3 of them alone, we could also combine them and create fun circuits. So just the way that many of you in tech are used to thinking about circuits and Boolean logic and these types of things, if and, we can do those same types of things with DNA. We can engineer in these logic circuits. If you experience this, do that. If you experience this other thing, don't. Right? So you can do that with DNA. You can do this with cells and viruses. And so we got excited about testing all of these payloads in combination with each other, and we just decided to make thousands and thousands and thousands of libraries of these viruses and then start testing them en masse. And sure enough, several of these just started to work like gangbusters, and we just got so excited at the potential of this. We started a company back in 2021, Siren Biotechnology, and that's when I resigned from my position to kind of bring this platform of these libraries of viruses that are now universal and can be used to treat many things. We're pointing the ship at oncology to start, but that's just where we're starting, and looking to see where we can take it from there, so.
Love it.
Yeah.
Mike, I've been hearing amazing things about gene writing. What is that?
So gene writing is a technology we're developing to literally write in the code of life, right? So we talked about the fact that DNA encodes our health, and unfortunately in certain circumstances our disease, right? So if one letter wrong out of 3 billion can lead to a fatal condition, or if a different variant could be protective of Alzheimer's disease in such a way that you stand essentially no chance of developing Alzheimer's through a normal, healthy lifespan, it stands to reason that if we could alter DNA, if we could restore it to its intended sequence, or if we could write it into cells to impart new functions, we could get to that vision of cure that we talked about at the start of this session. And so to build this technology, we look to nature, we look to evolution, I think, much in the same way that Nicole did, although we're looking at a different part of the equation. So we asked the simple question, has nature already invented the tools to write DNA? And the answer is that it has. And that answer lies in a family of genes called mobile genetic elements. You can think of them as nature's architects. They're evolutionarily ancient. They literally go back billions of years. And over those billions of years, they learned to do exactly what we're talking about, to make copies of themselves and write themselves into genome. And they're so successful at this that in this room, all of us, about half of our DNA comes from sequences that are derived from mobile genetic elements. And so we thought, can we tap into this power, and can we create a platform that allows us to make whatever change we need to address human disease, change any letter in any other, write a whole gene into the genome to restore function, or impart new capabilities like cancer fighting capabilities into immune cells. And by harnessing this power, we've built a platform that will allow us to do all of these things. And by coupling it with a focus on delivery as well, we're able to do this in living systems, and ultimately we'll do it in patients.
So a common point in both of your perspectives is that if you can find a means by which to intercept disease, which is modular, programmable, or held in common across many diseases, that if one of those works, it's likely to work across many. In fact, at the beginning of Moderna, a biotech company that has embodied some of these sentiments, they had famously said that there would never be a Moderna with one drug. It was either going to be zero or it would be a whole bunch. And that mRNA, being the center of the central dogma of life, was not going to work in some obscure corner of medicine only. Mike, I'd love to hear, how have you thought about developing the GeneWriter platform, and what are some of the points of evidence that lead you to believe that one cell type at a time or otherwise you're going to be able to navigate the continuum of curing disease to preventing disease from happening?
Well, as you said, Jeff, over the course of the last 20-some years since we've had the availability of the draft sequence of the human genome, we've learned a lot about our ability to read the code of life. And we know of literally thousands of conditions where a single gene defect leads to disease. Unfortunately, that's often life-altering or life-shortening disease. And so we have all of this knowledge, but we've had no way to capitalize on that knowledge. We've had no way to correct those errors to deliver cures. And so what we are doing is developing the technology that goes after conditions where the cause is unambiguously known. So sickle cell disease. We've known the cause of sickle cell disease for 70 years. There's just been nothing to do about it until very recently. If we can correct the single letter mutation that differentiates a patient with sickle cell disease who will die very early in life, in their 20s, 30s, or 40s in the developing world, probably before their teens, in the developed world, probably before their teens in the developing world, we can deliver a cure. So we have the cargo that can do that, and then we have to make sure that we can do it in living systems. So we have to find a way to get that cargo to the cell type of interest. And that's been a major focus of work at Tessera. And when we put those two together, we have a level of certainty in our ability to deliver a cure that doesn't exist really anywhere else in drug discovery and development. The most common reason for failure of a drug discovery program, and I've run literally hundreds of them across the course of my career, is that you had the wrong idea from the outset. You thought protein A was going to help disease B, but it just doesn't. Because the diagrams in your textbook, everything you read in the last edition of Nature was wrong or incomplete. But in the diseases we go after, we know unambiguously what we need to do, and then it becomes a technical challenge of can we do it, and can we do it in a living system? And if you can do it in human cells, in culture, if you can do it in a mouse, to move into living systems, if you can do it in a non-human primate, the likelihood that you're going to succeed in a patient is dramatically elevated compared to the history of this field.
So I want to come back to that because conquering sickle cell disease at scale would be an aspiration worthy of the entirety of a company, and yet starting where you began, if that's true, then Tessera's platform is going to be applicable to a lot more. But Nicole, I want to pull on the thread that you began. So you were seeking a means by which you could have a universal means of influencing important diseases to humanity. Many times people think of gene therapies as being for N of one, N of a few, just a few patients with a rare disease. How did you approach things from the other side of the spectrum?
So this was really kind of a perspective shift. If you were to go to a gene therapy conference, my views would be considered heretical, right? It's not a space that we talk about. It's not a space that we enter. Everything is viewed from this very monogenic lens, a single gene that has a single defect that's known, and the idea of coming up with a payload that could be used to treat more than one indication is just kind of a thought that hadn't really occurred to a lot of folks. And so I think the best way to convince any scientist is with data, myself included. And when we started this project of generating these universal payload kind of libraries, the way we convinced ourselves was actually making the libraries and then testing them in as many use cases as we possibly could. The classic example, I was actually talking with Jeff about this yesterday. We quite literally walked around campus when I was faculty at UCSF and just found collaborators on campus who were famous in different disease areas and disease spaces and had known model systems for those diseases and said, will you collaborate with us? We have these viral libraries. And actually seeing the data readouts from those, they're like, gosh, they keep working, they keep working, they keep working. That was really when we got excited and realized that we were probably on to something. There's a reason, it's like a classic example here. Every single one of you in this room, not to freak you out, every single one of you in this room has a tumor. You have had a tumor, for the most part, every day of your life. But it's a single cell. It's not a big, huge baseball-sized mass. So you've got one in your leg, and you've got one in your kidney, and you've got one in your heart, right? They're all over the place, but they're single cells. And your body knows how to fight and kill all those tumor cells. It's been doing it every single day of your life. How does it do that? Right? Those individual cells send off all kinds of little chemical cues and signals to the local environment, and the healthy cells that are around it can sense those and pick those up. And they can create little alarms, little beacons, little sirens, hence the name of our company, and tell your immune system something's up with this cell. It smells fishy, it looks fishy, it's acting fishy. Come and do an investigation. And your immune system will come, run a little investigation, and be like, I agree, something's up with that guy, and they'll chew it up and eat it while it's still a single cell. And so that's how you are able to fight cancer every day. Your body already knows how to do this. This is the concept behind a field called immunotherapy, where you reinvigorate your immune system or deliver new cells to reinvigorate your immune system, to reteach your body to do what it already knows how to do. And so we can do this, we can now do this with viruses, where we can use a virus to go and tell your immune system, come run this investigation, something's funny with this guy, and they can come up and chew up the cancer cells that have evaded your immune system, and that's how they were able to kind of create that baseball-sized tumor. And you can reuse and kind of retrain to kind of play off what Mike was saying earlier about, like, nature already knows how to do all of this stuff. Your immune system is no different. Your immune system already knows how to fight many of the diseases you have, but sometimes it can get overwhelmed, and you need to kind of reteach it how to do what it already knows how to do, and so we're kind of leveraging that in a new and interesting way.
Mike, I want to bring this back into the therapeutic formats that Tessera uses. So in a handful of cases, there have been miracles in editing cells outside of the body, painstakingly bringing them back in, and patients have been cured of cancer, or a subset of patients have been cured of diseases like sickle cell. Unfortunately, those don't scale to the level at which the patients today could benefit from those, and some of those regimens are as or more expensive than the medical system is willing to reimburse. Everything Tessera is doing has the opportunity to turn that upside down. How do you think about the scalability if the technology does what you described?
Yeah. Well, we at Tessera exclusively pursue in vivo therapies, so therapies that are delivered to patients and work within the human body. And to date, some of the largest successes in what I'll call genetic medicine, even though people don't think of it as genetic medicine in the application that I'm talking about, have come in cell-based therapy, and they've come by removing cells from the body. And there's a very simple reason why those are where the successes have been. It's the only place we could do complex manipulations. So you can take cells out of the body, you can do complex manipulations in a petri dish, if you will. It's not really a petri dish, but we'll call it that. You can manufacture it for a month under artificial conditions and try to put it back into the body. But there's a couple of problems with that. There are very, very few cell types that are amenable to that kind of manipulation. Basically, our white and red blood cell systems. Secondly, it's extraordinarily cumbersome to do that, and to re-administer those therapies, it basically takes a stem cell transplant, what we traditionally call a bone marrow transplant. That is an enormously challenging procedure, right? So the first step in receiving a stem cell transplant after you harvest the marrow is you give what is a lethal dose of chemotherapy to the patient. Without rescue, that patient will die. You eliminate their white and red blood cell systems. Then you rescue them, and there's a reason why we call it rescue with the marrow that you have manipulated. There are patients who, for example, with cancer, have received cures, and it's a wonderful thing to cure any patient with advanced cancer. But the ability to scale that approach, even in cancer, is just not practical. It's just not feasible. And when one tries to think about conditions like sickle cell disease, the ability to do that across millions of patients is just a logistical roadblock. There's no way in the world that we could commit that many academic beds, because it takes academic medical centers to deliver these therapies, to make a broadly applicable cure for sickle cell disease. And that's in the US and in Europe. It's not even something that you can begin to think about in the developing world. And so we are developing the tools to make those complex manipulations, but to make them in patients. So instead of that stem cell transplant that I described, think of it as an IV infusion. You go in to see your physician, maybe you go into an infusion center, you sit in a chair for a few hours, and then you go home. That is night and day different from what I just described, and it's what we think will be necessary to unlock the real potential in these genetic medicines. And so having that sort of scalable cure for sickle cell disease, scalable cure for advanced cancer, is one of the major goals of Tessera.
I want to pull on that word. So cure has been a term that has been a quiet aspiration for the entire history of medicine, but in fact, there's a lot of hesitation to utilize it. The simple rationale for that is probably obvious. It is rarely achievable in medicine today. We started with the reasons to believe that DNA is going to change that in the long run. I'd love you each to end with what does greatness look like in the coming several years for your endeavors? Nicole, please.
I would love if the universal gene therapy actually panned out and we were able to take a single one of these universal gene therapies. And again, there has never been one of these before. So if we had one that was able to work for one thing and then we were able to get it to work for a second and kind of prove out that platform, prove out that concept, and then now it's just off to the races. How quickly can you raise capital to just go after everything? So then it just becomes a capital play and not really a scientific play. And I think if you start to actually cure not just one thing, but two things, the money will flow. So I'm hoping that we can actually get to a place where it starts to become, hopefully, more of a kind of cookie cutter problem. Now it's just like, how quickly can we copy, paste, find, edit one IND? So that's the packet that you submit to the FDA where you ask, can I run a clinical trial with this drug? Can we do copy, paste, find, edit, and just next disease, next disease, next disease, and just start going down the list, and it becomes formulaic. So that's the hope. It gets boring, it gets formulaic, because it becomes so easy.
And what's the first chapter of that? Is it oncology? Like, walk us into patients today that may benefit.
So I think the first chapter for us, at least at Siren, the first chapter is going to be oncology, and we're actually starting in the brain. Why the brain? So the brain is kind of the Wild West. It's our white whale in the cancer world. It's just a tissue where we just have nothing. There's about 126-ish, depending on which physician you ask, different cancers in the brain. There are no effective therapies for any of them. None. The worst of these cancers can kill you in about 6 months. The best one of these, not that there is a good one, but the best one of these is single to maybe double-digit years. But the worst of some of these are literally months. These are single-digit months that you have once you get one of these. And so there's really, the best market to go into is one where there's no competition. And so when there are no effective standard of care and the needs are really immense, the brain is also a really interesting tissue because your immune system doesn't get up there. Those red and white blood cells that Mike was just talking about don't get up into your brain. You've probably heard of the blood-brain barrier. So there's this huge opportunity in the brain space and not a lot of competition. So, I was of the opinion if you're going to do something big, swing for the fences. So I didn't leave academia, the really safe, cushy, Ivy Tower, to go do something small. So we wanted to start in the brain since the need there is immense, and we'll move on from there. But we're starting in the brain. So.
And Mike, your medical background is one of the things that I've loved. The incision with which you've imagined changing medicine has been spectacular.
Yeah, well, I'm a physician scientist, as I said, and I was originally an HIV immunologist. In addition to doing basic science research, I took care of HIV patients in the early days of the epidemic. And I've had the experience of sitting across from a patient and basically telling him or her that there's really nothing that we can do for them and describing what fate lay ahead of them and trying to do the best I could to counsel them, but feeling just completely inadequate in the ability to do my job and to provide them a real benefit. And so what I think success looks like is when a patient who's facing life-shortening disease, when the parents of a young child with sickle cell disease can come into your office and you can tell them, it's okay, there's something we can do about it. Or any one of the countless conditions that we're trying to treat that are either rare genetic diseases, that are advanced cancers, or over time common diseases, because there's no reason why the approach that we're describing can apply to common diseases as well, conditions like Alzheimer's disease. When you can change that from counseling patients about what's ahead to telling patients that it's okay, there's a cure. It's night and day. And we can talk about the ability to drive success. We can talk about the ability to build great companies. If you can do that, if you can do what I just described, you will build a great company. You don't have to worry about anything else. That essentially is the core of what genetic medicine holds for the future.
Beautiful. Well, join me in thanking our panelists today, and thank you for your interest and attention.